Good morning, everybody. Sorry for being a little late. I had some other commitment. Today we're going to discuss the stem cells in the cardiovascular system. Not like the stem cells, the adult stem cells we have covered so far, which is the stem cells in the small intestine, in the hematopoietic system, in the skeletal muscles, and some cells in the bone marrow, like the mesenchymal stem cells. So all the stem cells we have discussed so far, more or less showed a lot of evidence, strong evidence pointing to the existence, the identity of those stem cells as well as repetitive demonstration of their functions. A lot of those were demonstrated at single cell level. So a lot of them were shown to be able to repopulate the organ with just one cell. I hope you can appreciate at this point that to demonstrate the identity of adult stem cells, the best experiment is to show that a single cell can repopulate the intended differentiated cell lineages in a given organ. But today, in the cardiovascular system and in the next lecture, but I would say, especially today, there are a lot of debate whether there are stem cells in the cardiovascular system, as well as, if there are stem cells, different groups have proposed different, populations of stem cells in the cardiovascular system. How those population of cells related to each other is still a matter of debate, because there must be more than 10 different stem cells described so far. I guess I'm being conservative to say more than 10. There are a lot of stem cells described in the cardiovascular system, but how they are related to each other. Are they the same cell, or they are functionally related? There are just so many different kinds of stem cells in the cardiovascular system. It is a matter of debate. So here, you won't hear a lot of certainty. It's still ongoing research, a lot of debate going on. That's just something you need to keep in mind. The knowledge is still not, we cannot call it knowledge yet. It's still ongoing research. But first of all, I would like to describe the power of the cardiovascular system. And before that, I would like to introduce to you the two different circulation. One is the circulation in fish. The other is in human beings or a lot of other mammals. In fish, what we describe the circulation system is a gill circulation, because they don't have lungs. They have gills. And then usually the heart is not a four-chamber heart like us. The human being have left ventricle, left atrium, right ventricle, and right atrium. So it's four-chamber. But in fish, you have only two chambers. Although from the illustration, it looks like you have three chambers, but actually we call it just two chambers. It's the same chamber. They will have to generate all the force required for the entire circulation. So first, the force will be able to pump the blood toward the gill capillaries. So in the gill capillaries, where you will find the exchange of oxygen, right? The outside oxygen will jump to the hemoglobin of the red blood cells, and now the oxygenated blood will go to the body's capillaries, right? And after that, the blood is pumped back into that same two-chambered heart. As you can tell, now the force is from the single chamber that will have to first go to the first capillary bed and then to the next capillary bed. So it has to work very, very hard, right? It's the single chamber that will generate the force that goes to the gill and then goes to the rest of the body. But in human beings, So you have a four-chambered heart. Not only that, but also the left and the right are separated. So the circulation in the lung and the circulation in the rest of the body are two systems. Okay, so from the left ventricle, you receive the blood from the lung, right? From the left ventricle, what you get is oxygenated blood. It's already rich in oxygen. And now the left ventricle will just pump that to the rest of the body, except the lung, right? To the rest of the body where it provided oxygen and other nutrients to for all the organs and body parts to function. And then from the venous vessels, now the blood get pumped back to the right side. Now from the right side, the right ventricle will pump the blood towards the lung capillaries, where you get oxygen exchange. So you can see the right side is pumping the blood toward the lung, while the left ventricle is pumping the blood to the rest of the body. In the right ventricle, the blood is less oxygenated, right? It's oxygen poor. It pumps the oxygen poor blood to the lung to get oxygen exchange, and then the left ventricle will continue to carry the oxygenated toward the rest of the body. So that's the two different systems in fish and in human beings. But in either case, the heart is such a powerful organ there. So I have some numbers for human beings. Our heart, in average, will beat about 3 billion times. That's a lot throughout the lifetime. And then the amount of blood that will pump out for life is going to be 30 million gallons of blood. Imagine for most people, the heart is almost the size of your fist, right? It's not such a big organ there, but it's so powerful. And for most people, it's going to last for many years. From birth to death, it's going to be functional, without any problem. So you would think there may be some repairing, some maintenance going on, right? But as you will learn today, for the large part, we still don't know what is the maintenance system there, because we don't know whether there is stem cells or not. Okay. Because the heart is so powerful, and the heart itself also needs blood supply. It's no wonder that the myocardial infarction, which describes the shortage of blood supply to the heart itself, is the #1 killer for people in developed countries. So usually because of lack of activity, buildup of fat in the blood vessel, there is a tendency for people who are prone to blood clot to have their coronary artery blocked by clots, or simply by the narrowing of the vessels because of the buildup of fat along the wall of those vessels. Now blood supply toward the heart is not cut off. Now the heart cannot receive blood supply and oxygen and other nutrients. The cell that require the blood supply will die. The heart cell will die, and eventually the heart will fail. And once the heart will fail, the people usually will die within 5 years. So that once the people have a heart failure, their survival chance is lower than most of the cancers. So that's why myocardial infarction and the heart failure is the #1 cause of death overall, for most countries. This country, of course, included. So today, we're going to describe the stem cells, and hopefully you can learn something, although we don't have a good knowledge as in other systems, but hopefully if in the future you become a doctor or you become an investigator in biomedical science, you can use what you learned today to find some cure for the disease we just mentioned. So in addition to the heart, we have a lot of vessels. right? We have a lot of vessels that made up the circulation system. So we can classify the vessels to three groups, right? Artery, veins, and capillaries. So shown here, A is artery, B is the structure of veins, and C is the structure of capillary. I will start with the simplest. For capillary, essentially what you have is just a single layer of endothelial cells. Because you only have a single layer of epithelial cells and no additional structure, capillary will be used for the purpose of gas and nutrient exchange between the blood inside the vessel and the surrounding tissues, right? It's not much to say. It's just a single layer of endothelial cells. Now, move on to A, that's an artery, as well as in B. You can tell the vessel wall can be divided into three parts, mainly. We call it tunica interna, tunica media, and tunica adventitia. The three anatomical terms are used to describe, interna means the interior part, right? And media means the middle part, or the adventitia means the outside of the blood vessel, the outside layer of the vessel. So let's describe the three layers in a little bit details. Of course, the closest to the lumen will be the endothelial cell. You have just one layer of endothelial cells. That's the main part of the tunica interna. And of course, you have some basement membrane there to line between the tunica interna and tunica media. You have a membrane there. And then your artery, you have the thickest part, you have the thickest tunica media. You have the thickest tunica media there. Because, for artery, the pressure inside the artery is very high, right? Because the left ventricle is very powerful. It will put a lot of pressure to the arteries. And then the artery must be very flexible. So between each heartbeat, sometimes the pressure is lower, the diastolic is lower, and then systolic is the pressure will increase. So the arteries will need to be more flexible than veins. Therefore, you have a very thick tunica media, because the tunica media is composed of smooth muscle and elastic lamina, elastic fibers. So you can tell the smooth muscle is kind of contractile, sort of contractile. The elastic fibers are, of course, very, very flexible. That is made up that makes up the media part of the vessel. Okay, the outside, the tunica adventitia is mainly connective tissue, but you do have nerves in the outside the adventitia part as well. So the nerves are there as well. So if you compare the structure between arteries and veins, in veins you have valve. Now the pressure there is not as high. So the pressure is not high. So the valve serves as a purpose to make sure the blood flow at just one direction, right? And now, if you look at the tunica media, you don't see the elastic fiber, and also the smooth muscle layer is much thinner when you compare to the artery. I explained the reason, because it is less flexible. The tunica media is less flexible. The outside part is still the tunica adventitia. So that's the structure of the three blood vessels. As we will see, they receive a lot of stress, mechanical stress, especially when the individual is under some kind of stress, either physical stress or mental stress. Your heart is pumping much harder, and now that will cause damage. That will cause damage to the endothelial cells and also to the smooth muscle cells in the tunica media as well. So those two cell types will have a lot of damage. So people predict that there must be some maintenance going on. There, it may not it may not involve stem cells because we described that tissue regeneration. Tissue regeneration doesn't always involve stem cells. So cell proliferation sometimes can fulfill that purpose. Anybody have a question?
Yes. So I have a quick question. So as you just mentioned that stress can cause the damage of the tissues, right?
Yes.
And so is that like, and sometimes they recover and sometimes they don't. So is that meaning that like the mainly the heart attack that happen, is it mainly like if a person is under too much pressure, so is that the like mainly cause or mostly cause that heart attack happen?
No, I said it was both the physical and mental,
Oh.
especially when you have a, like a, I, I, you usually not chronic stress. If you're talking about mental stress, it's not a
Mhm.
chronic stress. It's rather something all of a sudden like an accident,
Mhm.
right? When that happens, your blood start to pump, your heart start to pump very very hard because of the adrenaline. Yeah,
Mhm.
in that case, the stress towards the vessels will be will be larger.
Well, okay.
Okay,
Thank you.
okay, it's not about chronic stress. Okay, so previously people think for many years people think that the major maintenance system is by the cells re-entering cell cycle. So before people recognize that there may be stem cells there, located within the vessel wall, people feel like maybe there is no stem cells, just those endothelial cells and the smooth muscle cells. That those are terminally differentiated cells, right? They don't usually enter the cell cycle. They don't proliferate because they just perform their function. But once there is some damage, for example, like a short of blood supply, or just or sometimes just because of, well, you know you work too hard, all right? Physical work out too hard, and it can cause damage along the some of the vessels. And now in damaging situations, maybe the endothelial cell and the smooth muscle cell will just re-enter cell cycle and start to proliferate. And now you don't need the stem cells. They will proliferate and then repair the damages. For quite some time, that's what people feel what's going on. But now, you know, uh, in the past 10 years or so, people start to recognize there may be stem cells within the vessel walls. So that's the current perception about maintenance, vessel maintenance. It's because of progenitor cells or stem cells within the cell wall. So today I'm going to describe some of those, uh, not experiments, some of the theories. I wouldn't say anything is concrete at this point. So just some of the experiments as well as the conclusions they have reached. So we're going to describe three main progenitor cell type. The first is endothelial progenitor cell, the other is smooth muscle progenitor cell, and then we discuss multipotent vascular stem cells. So as I said, nothing is is concrete at this point. So people proposed that there may be epithelial stem cells in circulation. It's not within the vessel walls, rather in circulation. So in those studies, what people would do is just to, uh, collect the blood cells, because, in a plasma, you just spin down the cell. No matter what is what is there, you just collect the cell, and hopefully some of those cells are not the hematopoietic cells, like a red blood cell, monocyte and such. You hope there are some other cells that are progenitors saved for the purpose of repairing vessels. You hope there are those cells. That's exactly what they did. So they feel those cells should be more adherent than the hematopoietic cells, because the hematopoietic cells are always circulating. They don't need to adhere to anything. But if there are some cells that are more adherent, maybe they will attach to the wall of the vessels and then start to repair the wall, right? So that's a very straightforward thinking. And then once they isolated those cells, they put into some approach like creating injury in some parts of the rodent body using an animal model, right? For example, like they will tie up blood supply toward the limb, right? So some vessels in a limb will have damages because they lost blood supply. The vessels are just like any other organs. They also need blood supply. So if you tie up the vessels there, you're going to cause damage in the endothelial cells and the smooth muscle cells. And now you have a disease model. And now you just inject the cells that you transplant the cells that you have identified. You feel that they are epithelial stem cells, and then you inject into the injured place and see whether those cells will repair the wound or not. That's a very straightforward experiment in there. So indeed, they showed that those epithelial stem cells identified this way can repair the damage there. However, there are a lot of concerns. Why? It is because even in the hematopoietic cells, like the monocyte and some of the lymphocyte, there are some population in the hematopoietic cells. They have function to help repair the vessel. So basically, it's very difficult to distinguish the true epithelial endothelial progenitor cell and the pro-angiogenic hematopoietic cells. So some hematopoietic cells have the function to trigger angiogenesis. Angiogenesis describes a process where you have extension of the vessels, because for example, like when you do exercise, your muscle become bigger, and your new muscle will need new vessels, right? So the vasogenesis is termed as term angiogenesis. Angiogenesis is always going on throughout our body. So some hematopoietic cells are known to promote that process. So it's already known that some cells in the hematopoietic system have that function. And now you isolated some endothelial progenitor cells, and now you have the burden to distinguish that these endothelial progenitors are different from the pro-angiogenic hematopoietic cells. It turned out to be a very difficult task because, as you can see, of course, you have to keep in mind that some groups of scientists describe the pro-angiogenic hematopoietic cells, and other groups that describe the endothelial progenitors. And now you have to put them together to compare whether they are the same cells, right? So here is a table that summarizes, the differences between these, or the common features of these two populations of cells, proposed by quite a few groups. As you can see, they share some markers like CD31 and CD34, and CD45 is kind of like more unique to the pro-angiogenic hematopoietic cells. But you don't have a lot of those markers, the cell surface markers that can be used to distinguish this so-called two populations of cells. So I will not go down the list, but the idea here is, although different groups describe the presence of endothelial progenitor cells in circulation, but it's hard to say that they are different from the pro-angiogenic hematopoietic cells.
Dr. Liu?
Yes.
So there's one problem I'm trying to understand. So pro-angiogenic hematopoietic cells are the ones leading to new angiogenesis, correct?
Yes.
But like the blood vessels that are formed have endothelial cells. So wouldn't they be basically also leading with endothelial progenitors at the same time?
No.
Okay.
Well, I guess what I'm trying to say here is when you, for example, in a cell transplantation, you
Yeah.
can transplant the so-called endothelial progenitor cells and demonstrate, okay, now the cells get incorporated into the new vessel, right? Or
Okay.
for the damaged part. Or you can inject this pro-angiogenic hematopoietic cells in there, but these cells are pro-angiogenic. Therefore, you're going to have new vessel formation, but now basically this promotes the recruiting of some endothelial cells towards the damaged place. So
Okay.
yeah, unless you have a very good linear tracking system in place,
Yeah.
you cannot distinguish whether the cells that contributed to that repairing is endothelial progenitor cells or just the other endothelial cells.
Okay, so what I'm understanding is that the proangiogenic are the ones that recruit the endothelial progenitors.
Yes, not
And-
themselves. They
yeah.
recruit other cells towards the damaged site and
Okay.
perform the repair.
Okay, so that makes sense because then the clonal proliferation and in vivo vessel formation is negative for proangiogenic because they're not the ones making the new blood vessels, they're recruiting, and the
Of
endothelial
course,
the ones
of
who are.
course.
Okay, great.
Of course, because these are rare populations. That's why, if you don't have it is extremely difficult to do this type of linear tracking. As you can see, they share a lot of common genes.
Yeah, yeah.
You may not identify a factor that you can use to perform linear tracking.
Yeah,
Therefore,
true.
whenever you inject these cells, you always have the problem of whether these cells themselves contribute to repairing or the cells recruit other cells.
I see.
You always have that trouble if you don't have a linear tracking system.
Yeah, that's really difficult.
Okay, so that's why up to date it's still a debatable matter whether you have endothelial progenitor cells in circulation. So we don't have a final conclusion yet. Now on this slide, I'm going to describe endothelial progenitor cells within the vessel wall. So I would say this is the more dominant theory at this point. People feel like the progenitor cells, endothelial progenitor cells, are not in circulation. They're rather, within the vessel. And people have described their phenotypes, or in other words, their the markers that you can use to identify those. As you can see here, again, it's a CD34 positivity, but you must have some other markers such as VEGF receptor positivity and Tie2 positivity. Basically, this VEGF receptor and Tie2 are specific markers for endothelial cells. So these are usually not found in hematopoietic cells. So within the vessel wall, you can isolate some cells that bear more identity of endothelial cells. Okay, so they are progenitors, but they bear some marker of endothelials. In contrast, probably they don't have the markers of hematopoietic cells. So that's one example. From one of the study of human thoracic artery, you can isolate the cells marked by these three extracellular markers. Now I will jump to the third example. The people have isolated from a lung vasculature of mouse. They use just antibody. Okay, use antibody to detect four markers here. You can see it's CD31, positivity CD105, SCAR1, and KIT. Why I bring this one up as the example is because I want you to appreciate that although we're discussing stem cells from different systems, you can see the markers sometimes are common, right? The SCAR1 and KIT positivity were first described in hematopoietic stem cells, right? Remember I believe it was lineage negative, SCAR1 positive, and KIT positive was the marker used for long-term hematopoietic stem cells, right? And now you can see some of the markers are used for the isolation of other adult stem cells. Actually, both SCAR1 and KIT and the CD133, there are common markers frequently found in stem cells, adult stem cells across many tissue types. So I will describe a little more detail about the experiment, kind of validating the stem cell or progenitor cell identity of the cells isolated from mouse lung vasculature. So here is the experiment. Why I chose this one to describe is because this one provides the the evidence that lead to maybe the best conclusion or the best evidence for a conclusion that this may be stem cells. Okay, so what they did is just they first perfused the mouse with a fluorescence dye. With that perfusion of the fluorescence dye, all the functional vessels will be labeled. So they label the recipient with a dye, a red fluorescence dye. So you know any vessels that's functional now will glue red fluorescence. And now they will inject this CD31, CD105, SCAR1, and c-KIT quadruple positive cells into the injured site. They create an injury and inject the cells to the injured site. And now this donor cells are labeled by green fluorescence by some genetic tools. You can always put the GFP under a universal marker so that the cells will be all green fluorescence positive. So they are selected by the 4 surface markers, but they also have an additional marker that's green fluorescence. And now they observe where the cells will ended up with. Okay, so this is showing you, a cross section of one vessel. Okay, the cross section of one vessel. As you can see, green fluorescence positivity shows that the donor cells contribute to this vessel structure. Okay, you have a lumen there, and that's the vessel wall there, and you can see they use the CD31 to label the endothelial cells. CD31 is an endothelial cell marker. Okay, even mature endothelial cells will express CD31. So they identified that the injected cell contribute to the structure and become CD31 positive cells. That means they become endothelial cells. As you can see from Debye staining, there are about 4 cells for this structure because you see 4 nuclei there. So when you have everything merged together, the conclusion here is when you inject this quadruply selected cell as a single cell. That's one thing I forgot to mention is they injected just a single cell in the injured site. And now that single cell can become endothelial cells and become functional because only the functional vessels will be possibly standing for the red fluorescence. So this is a good demonstration that these quadruply positive cells fulfill the criteria we discussed so far. That's a single cell that will be able to contribute to a structure. But I still want you to appreciate the difference. As you can see here, we are not using a lineage tracking system such as we used for the in testing stem cells. In that place, you just label a cell at the stem cell niche, and then you observe how that, over time, in real time, almost, how they contribute to, to all the cell population in that crypt structure, right? Here, you're kind of describing, you isolated some cell, you put them into culture, and then you put a single cell in this place, and you showed maybe your cell get incorporated into an existing structure, because you can't tell whether that is a new structure or it's a structure already there, but you can show that your cells contribute to that structure. You see the difference here, right? Once in vitro, they're done in vitro followed by in vivo, while the other is always in vivo. And also this in this experiment, your cell contribute to a structure. Well, in the small intestine stem cells, what's shown is one cell that contributes to the entire structure. Okay, that's the difference there. That's why this is still under debate, whether you have endothelial stem cells or not. It's still under debate because one argument is, well, you isolated those cells, you put them into culture, maybe those cells get spontaneously reprogrammed toward a stem cell, right? You cannot, this is like a very harsh argument there, but you cannot exclude that possibility. Although I would say that possibility is low, but that's still a possibility. But if you do it everything in vivo, for example, here, you isolated the cell from the vessel wall. So why can't you label the cell and now show during a damage how this cell contribute to the damage, or right there around the stem cell, right? So hope you appreciate a little bit about that.
I do have a question.
Sure.
So could it be that you for in terms of a blood vessel versus the intestinal wall? I remember you mentioned that intestinal walls can change completely, I believe, every 5 days, was it?
Yes.
And so could that have something to do with the fact that lineage tracing would be required for intestinal epithelial cells versus blood vessel walls?
Yeah, I appreciate what you said. Basically, there are a lot of regeneration in the small intestine, but not as much regeneration going on in the blood vessel. Therefore, it may not be easy to demonstrate this with the same approach here,
Exactly.
but it's still
That's what I was
possible.
thinking.
But it is still possible if you have a marker to label the cells, and then you just cause a widespread damage. Yet I'm sure the stem cells will not be located in just one place. It should be in many places. If your lineage tracking can mark those all the stem cells, and now you just observe in multiple places and see if you have the luck, to identify some active repairing, right?
Yeah.
Not necessarily to replace the whole structure, but you can observe the progression of
on a wider
repairing
scale
from the beginning to the end, right? But as you can see from that experiment I showed you in the previous slide, is not the case.
Right.
Okay, so that's why they still have more work to do to prove that point. Okay, but a very good question. And now let's move on to the multipotent vascular stem cells. That we say this, although people say this is multipotent vascular stem cell, but it's just a very strange term. People say this multipotent vascular stem cell, but when we see this term, usually we feel like this will contribute to the smooth muscle cells. So it's just because this in a lot of confusion in this area of research. So sometimes you see a term, but it's hard to explain from just the face meaning of that term toward the real meaning. So when we say multipotent vascular stem cells, you mean these cells that will contribute to the smooth muscle cells.
I do think that.
Okay, it's just a lot of study are going on. So when you see a term, what's the real meaning? You can memorize that. But the dogma here is again different from the endothelial progenitor part, because people have known for quite some time that there are two types of smooth muscle cells in the medial region of the vessels, and one's called contractile vascular smooth muscle cells, while the other is called synthetic vascular smooth muscle cells. People recognize this. There are two types of cells within the medial part of vessels for many years. So basically, these two cell types are different because one, by the name, is contractile. They're functional. Well, the other, I don't know whether they call it synthetic, but those are less contractile. They don't have perhaps don't have the as nice structure as the contractile smooth muscle cells. Therefore, their contractility is not as good. But if you study the gene expression, you will see that in the synthetic vascular smooth muscle cells, you have genes responsible for cell proliferation and for cell migration are expressed higher. Well, in the contractile vascular smooth muscle cells, of course, the contractile genes, the myosin heavy chain, myosin light chain, those are responsible for contractile ability will be expressed higher. So the dogma for many years is these two cells can change to each other. Basically, when there is injury, these contractile cells will become the synthetic vascular smooth muscle cells, and those cells will proliferate and then give rise to all the maintenance and repairing. So that's the dogma. But it's just like in epithelial progenitor cells. Recently, the last 10 years or so, people start to recognize that there may be progenitor cells or stem cells right there within the vessel wall. Okay, it's not just the contractile cells that re-enter the cell cycle. It's some unique populations there. But what are those unique populations? There remain a lot of debate. So I'm not going to go into the details because it's probably even more confusing than the epithelial progenitor part. But I just want you to memorize what I said at the beginning. They say from the three part of the vessel, like the intima, media, and adventitia, you can isolate some special cells that are called multipotent vascular stem cells, and those can give rise to synthetic smooth muscle cells. So, you try to integrate the new theory to the old theory, so you call the smooth muscle cells derived from these progenitors as synthetic SMCs. Okay, synthetic smooth muscle cells, and then at the same time, these stem cells can give rise to other cells around the vessel, and that will help perform vascular repair. So for the other cells, from some of the studies, they say these cells can give rise to synthetic smooth muscle cells as well as the endothelial cells. But not every research demonstrate that. So most of the research will demonstrate that the multipotent vascular stem cells give rise to smooth muscle cells, not endothelial cells.
Dr. Liu.
Yes.
So I was also wondering, because I think I know for atherosclerosis, when there is lesion to the smooth muscle cells, they change from contractile to synthetic, as you mentioned. But the thing is, it keeps being synthetic. It doesn't return back to contractile. So I was wondering, is it like a positive feedback generation in a sense?
Well, if they become synthetic, that means they are less functional.
Yeah, exactly. And that's the problem with atherosclerosis. So it's hard to return back.
I think maybe that is part of the reason why it's less flexible, become more and more rigid, and people will start to experience high blood pressure.
Yeah.
The high blood pressure is because of that, because the hardening of the vessels.
Yeah, and then that causes more pressure.
That may explain it, because it's the synthetic ones are maybe just occupy the place and then secrete some factors, but in terms of contractility, they're not as functional as the contractile ones.
Gotcha.
Yeah, possible. I'm not so familiar with that part, but I think that's a possibility there. But I'm going to describe this parasite because this is somewhat related to today's content, because remember, from the parasite, I'm not trying to tell you that parasite contribute to the repairing of the vessels. They may do that, but I don't think the evidence is strong enough. But along the vessels, there is this special cell type called parasite that is closely related to mesenchymal stem cells. Mesenchymal stem cells is, as I said, it's the most debatable in terms of their identity, in terms of whether mesenchymal stem cells from different organs are the same cell or are different cells. So it's a lot of debate going on. So I would say the mesenchymal stem cell field have good science and bad science. some fake science altogether. But the parasite is a cell type that's very interesting. There is some possible relation between parasite and the identity of mesenchymal stem cells. But first of all, I would like to describe what is parasite. As shown on this image, you see maybe some structure, some vessels within the brain, because you see a lot of neurons, astrocyte, and such. So those are usually in contact through some protruding part toward the vessel itself. Shown here is apparently the capillary. It's not a vein or artery, because you have just a single layer of epithelial cell and the basement membrane outside of it. But outside of this membrane, basement membrane, you see this red colored cell, which is called a parasite. Based on the, by the name of it, you can kind of predict that it is this some cell type around that vessel. It's on the outside layer of the vessel, and somehow it take a unique shape to kind of embrace the vessel. Okay, the nucleus is shown in a thick part, and otherwise it's very thin. The cell is very thin, just to embrace the vessel there. So this is called parasite. So people propose that maybe parasite is a major source of mesenchymal stem cells outside bone marrow, because maybe even within the bone marrow, because no matter you isolate the parasite from fat or from marrow or from all kinds of places, you always have a lot of capillaries in those organs, and then you have a lot of parasite. So people feel like maybe most of the mesenchymal stem cells are from parasite. It's not from anywhere else, it's from the parasite. So the reason is because if you isolate the parasite, now you're not just taking a fat and then make a single cell suspension. Now you start from vessels. For example, you get the fat tissue. Now you remove all the other cells, just leave the vessels behind, and now you try to isolate the parasite. And after doing that, you put the parasite into culture. Now people show that they can differentiate into adipocyte, chondrocyte, and osteocyte. So essentially, that's the essence of mesenchymal stem cell. So that's nowadays still under debate whether parasite is what gives rise to mesenchymal stem cell. Now here I have a table that shows the comparison between these two populations in terms of a lot of markers, surface markers as well as internal markers. As you can see, a lot of markers are shared, especially CD44, 73, 90. The markers people used to fax sort out the mesenchymal stem cell. So, whether they're related, there is a hierarchical relationship between these two cell population will still be explored, before we can draw a conclusion. It's just not so much related to vessel repair, but just a special cell type that can contribute to that may contribute to mesenchymal stem cell. So lastly, I'm going to use 2 to 3 slides to describe to you the stem cells or progenitor cells in the heart. First of all, I would like to point out that only about 10 years ago, people start to appreciate, start to develop tools to study whether you have cell renewal, cell replacement in the heart. One group did a very creative work. What they did is they know that around the 1950s and 1960s, between probably from 1955 to 1965, somewhere around that, the world, China, U.S. and the previous USSA, they all tested their nuclear bombs. Therefore, you have more radioisotope in the air, in the environment. So they take advantage of that fact, and then they use carbon dating to show, whether you have cell replacement in the heart. Now, how exactly they did that, I don't know, because it's not it's not common carbon dating. In biochemistry, you learned that you need a half-life to do a carbon dating, but in this case, you don't need half-life. But I can tell you one thing is they have shown people born before 1955, when the heart were analyzed for the presence of isotope carbon-14, they found actually the carbon-14 was higher than the environment. What does that mean? That means maybe the heart cell does have replacement. Otherwise, when those people were born, that's before 1955. Okay, the radioisotope level in their bodies are actually lower. And now, in the heart, you just analyze the heart. Now you have more isotope in there. That means the cells does incorporate carbon-14. Okay, so I believe what they analyzed is nucleic acid. What they show is, okay, you have more nucleic acid in the mouse. That means you have cell replacement. Otherwise, how can you have more, right? I think that's a good argument. The exact method I don't know, but they were able to calculate that. Please pay attention to the first row there. They say that there are about 1.5 per year of cell replacement in young adult humans. And that number will drop to 0.45 at 75-year-old. So although you have a very, very low rate of replacement, but it is possible. That's what they are saying. Some physicians said that maybe it is very significant, just that low level of replacement. Maybe the damage level is low, therefore you don't need a lot of replacement. So we still don't know. But I actually skipped some content from this chapter, from this point. The reason is because I unfortunately was in this field, in this cardiac regeneration field since maybe the beginning of my career in US. I was in a lab that was considered one of the pioneers who proposed the work of, who proposed the theory of adult progenitor cells in the heart. But another lab was much more famous. That lab is led by Piero Anversa. And for 10 years, he was the godfather of this field. But in about 2013, 2014, Harvard Medical School, his institute, said that he and his colleagues fabricated a lot of results. So Harvard Medical School asked them to retract almost all the papers they published over a span of 10 years. And it was terrible, and it was sometimes hilarious. I was in a meeting, when people challenged the work from that lab, and somebody just go up to the podium and said like, well, your work is excellent. From today's talk, your work is so excellent, but I have been following your work. I'm doing the same thing for the past 2 or 3 years. I just couldn't reproduce what you said. I did everything according to what you have described, but I just couldn't, reproduce it. And then there's a follower of that professor, Piero Anversa. There's a follower of that professor. And then this guy stand up and said, like, you know what? I like opera. I always try to sing like I'm Placido Domingo, but I can never succeed. So I guess if you cannot reproduce his work, that just means you're not good enough. That's pretty much not word by word, but that's what he said. But eventually, 5 or 6 years later, it wasted a lot of resources. You see, in high impact journals, people publish their work just to show that, Piero Anversa was wrong. They published some really high impact papers, but I would say, just prove somebody's wrong. It's really not as productive. I mean, for the professor who pursued that line of research, but eventually, that series started to die down. That's why I didn't even include that in this in this presentation. But our work that is SCA1 labeled cardiac progenitors still stand. So that's the work from the lab I was originally from. The work still stand, but the c-KIT positive stem cell led by Piero Anversa is now, people just don't feel don't feel comfortable that represent a stem cell population in the heart. But on this slide, what I want to tell you is something concrete, something solid. The outside layer of the heart is called epicardium. It's always known that when the heart is damaged, you are going to have cell migration from the epicardium toward the heart. Okay, during development, that's what happens. The cells first goes to the surface of the heart, the epicardium, and then they migrate into the heart to become cardiac myocyte and vessels, smooth muscle cells within the heart. That's what happened in embryogenesis. But if the heart is injured, you see the same pattern. The cell migrate into it, not necessarily creating cardiac myocyte, because usually if there's a damage in the heart, you have a scar formation. You don't have cell replacement, but you do have migration of cells from the surface toward the heart. So now these days, people are trying to take advantage of that observation to see maybe you can reprogram those cells. You can reprogram the cells that were migrating to the heart during damage. If you can reprogram those cells, the epithelial cells to become muscle cells, heart muscle cells, maybe you can make the heart to get improved in terms of cell replacement. So I ran out of time, so please read this by yourself. People describe that the cell migration could have two kinds of effect. One is cell replacement by themselves. So the cells from the epicardium can replace some of the damaged cells, for example, cardiac myocyte, which is still theoretical, right? Because nobody has proven that in a damaged heart that the epicardial cell will become cardiac myocyte, the muscle cells. So that's just a theory at this point. But additionally, those cells in the epicardium can release paracrine factors, that's growth factors that will diffuse into the damaged heart and maybe help the heart to get recovered. So we have a student-led discussion this Friday. They're going to describe how the cells, the fibroblasts within the heart, can get reprogrammed toward cardiac myocyte. Therefore, the heart will have a better function, after that kind of reprogramming in vivo, right there in the heart. And although the authors haven't proved that the fibroblasts were from the epicardium, but I think that's probably the case. So in the future, maybe that's one way to repair damaged heart. That's reprogram the cells from the epicardium. Until, of course, if someone of you or me, we can identify there are true progenitor cells or stem cells in the heart. Okay, that's the end of today's lecture. Thank you.
That's a really cool research question there, actually, because that would help a lot for cardiac injury.
Yeah. Because that's the #1 killer there. Yeah, we don't have any clue of cell replacement at this point. So that's why I feel I should point that out. You do have the epicardium naturally in development that contribute to the cells in the heart. And now during injury, it's shown they can migrate into the heart. They can contribute factors and cells that will go into the heart. And maybe we can take advantage of that road. To do something. In addition to identifying stem cells in the heart.
Yeah. Because
we're already doing cell identification for many years without much success.
But the first thing is that you can do maybe transdifferentiation to test it out, no? In a sense.
Well, in this case, that's pretty much transdifferentiation. We're just using two different terms, right?
Yeah.
You could call it transdifferentiation or cell reprogramming. It's essentially the same.
Yeah.
Right? If you're introducing factors to make one cell to become another type, you call it reprogramming or transdifferentiation. It's just different terms.
Yeah. So then how could you then identify for sure that the epicardial cell, like would you mark it, and then if it enters the heart following cardiac injury, then you would be able to identify that certain route? Is that
I'm
how
sorry, can you repeat your question again?
Yeah, so basically when you mentioned like it's still a theory, is the problem what is the exact problem of why we can't solve it out? Why we can't solve that question so easily?
Uh,
Like
what
why
theory? Can you remind me what theory I said?
yeah, why can't we figure out for sure as of now yet that the epicardium goes into
Well, now I understand your question. I feel, you know, if they contribute to myocyte, the cardiomyocyte in the heart, the number must be extremely low. So if you do a linear tracking, you may not be able to tell that. To tell that from artifact.
yeah.
Yeah. So basically, I guess people have done that already. Have done that linear tracking.
Okay.
To observe, you know, what cells will this epicardium cells become once you have an injury, because they do migrate into the heart. But I guess they seldom see that those cells become cardiomyocyte. That's why I say they keep that theory alive.
Yeah.
Okay, well, that's why I say it's theoretical, because they feel it's still possible for those cells to become cardiomyocyte naturally, but there's no evidence yet.
Yeah, and it would make sense that like it would be if it did happen, it would be so little because I mean people don't recover from heart attacks, so it's not that efficient. However, we could amplify that effect by allowing for epicardium to go in to enter that route. Correct?
Yes. I think that's a possibility. It's some hope there.
That's really cool.
Because in the heart there are a lot of fibroblasts.
Yes,
maybe
exactly.
more fibroblasts than the cardiomyocyte.
Yeah.
So what's the function of those fibroblasts in there? And now, like we discussed the other day, maybe they are more related to inflammation and the immune interaction of immune cells toward the heart muscle cells. That's maybe for some metabolic reasons, but there is a possibility of converting some of the fibroblasts toward cardiomyocyte.
Okay.
So you'll hear the student presentation this Friday. That's exactly some professors did.
Looking forward.
But in that work, they didn't show that the cells was from the epicardium. It just to show those are fibroblasts to begin with.
Okay.
So you begin with fibroblasts, you introduce factors into fibroblasts, and eventually the fibroblasts become muscle cells.
All right.
Yeah, the challenging part of both skeletal muscle and heart for those to proliferate is, they have a very organized structure, the sarcomere.
Yeah.
And a lot of times those cells are binuclei, quadrinuclei. So, for them to proliferate, it's very hard for them to overcome two obstacles. Once, evenly distributed nuclei, right? Because this is not like just one nuclei replicated two, and then you separated the two nuclei. They have multiple nuclei. How to properly separate those evenly into daughter cells is I guess is one obstacle. The other is you will have to dissolve the structure.
I see.
You have to dissolve the structure because you have a sarcomere there. The sarcomere is such a well-organized structure.
Yeah.
If you disrupt it, you're going to kill the cell, right?
Yeah.
You're kind of muscle inside the cell. The muscle is one muscle fiber is one cell. So now you want the cells to proliferate, you have to dissolve the muscle structure first. So you dissolve it, and once they form two daughter cells, the muscle structure will have to be rebuilt again. So it's kind of very difficult.
Yeah. All
So
right,
that's why people still thinking about progenitors, and then convert other cell types.
Yeah.
Yeah, they stay away from making the cells to proliferate. Not like for endothelial cells. Endothelial cells are much simpler.
Yeah, that's true. That's true.
Thank
Thank you so much, Dr. Liu.
You're welcome.
Oh, Professor, just out of curiosity, is your lab currently working on this kind of research with epicardium, or is this just a long time ago?
That's quite some time ago. That's quite some time ago because
Okay.
this area of research, because of Pierre Amertha, now become very difficult.
Because it's set everything back, right? All
Yeah,
the
because
research is
people, you publish a lot of great papers in all the great places, but one day people say, oh, those are all fabricated. So if you do similar work, you propose some research, it will say, well, you followed somebody.
so
that
basically
all
all
the
your
theory are wrong.
findings,
Yeah.
they have
So
to be
you couldn't get the support from community. So you better you do something else related.
Yeah, that's really sad.
Yeah, not exactly the same thing. Yeah, because that's very bad. So because a lot of researchers in this field, they don't have any fault, but they bear the blame.
Okay.
Yeah.
Thank you, Professor. This was such an interesting lecture.
Okay. Thank you.
Thank you.
Bye.